Morning, guys. Dr. Gillard here. This is Tuesday morning's endocrinology lecture. And yeah, we were talking about aldosterone the last lecture, and let's keep talking about aldosterone. It's a very busy molecule. We're going to get into it a little bit in this one. Aldosterone in the kidney. So aldosterone, of course, has effects elsewhere in the GI tract and on the heart, has negative effects, but its main target is to bind to the kidney and cause, well, we'll see what it causes. So aldosterone is a hormone that circulates, reaches the kidney. It doesn't circulate naked. About 60% of the time, it's actually carried by albumin. 35% is free time. Is just kind of free in there. Then there's other carriers it can ride on other carriers. It also reaches other important tissue, as we said, the intestine and colon. We'll talk about that. Sweat glands, salivary glands. We'll talk a tiny bit about that one. But its main target is the principal cells of the nephron. So they're found mainly in the collecting tubules. A couple AKAs for these collecting tubules. Guyton calls them the late distal tubules, or late distal convoluted tubules, or I've heard distal convoluted tubules. It's also in the proximal part or the cortical portion of the collecting ducts as well. And many authors, maybe the board question or my question might just say the distal nephron. So that's a less precise way, but it's certainly the same. Let's take a look. Remember our nephron. There's our loop of Henle. You can always tell the nephron by looking at that. You've studied this extensively. Bowman's capsule here. We've talked about the macula densa and the juxtaglomerular cells. And here's the potato pickers live here, right? This is the proximal portion of the proximal convoluted tubule, loop of Henle. But here's the part where the principal cells live. So it's really the distal last part of the distal convoluted tubule. But they give it a new name. It's called the late, Guyton calls it the late distal tubule, leaves off convoluted. Sometimes it's called connecting tubules. Several physiology authors just call it that. This is where the principal cells live. What's the response to aldosterone binding on these principal cells? Well, aldosterone binds to mineralocorticoid receptors, MR receptors. They're located on the principal cell, specifically on the basolateral surface of the principal cell. And this binding causes a bunch of stuff to happen. The first one we'll talk about is ENaC channels. So the binding increases the genesis of these ENaCs, or ENaC channels, epithelium sodium channels that live on the luminal or apical part of the cell membrane. Remember how the cell is shaped? Remember, if this is let's say where this is the distal convoluted tubule here, and we have a principal cell, we'll draw in, how about red? Right, so the filtrate is inside here. So this part down here of the cell, that's called the basolateral surface. Why don't they just call it the base? I don't know, but they do call it the distal or the basolateral surface. And then that's in very close proximity to capillaries. Right, here's a capillary, cartoon of a capillary right here. And so this is the spot in the middle. That's the interstitium. So we can get sodium and water going this way, right? Well, who goes the other way? Goes that way, into the filtrate. Good. Potassium. Who else? Potassium. Good. Hydrogen ion. So sodium, water, the effect of aldosterone binding. We could say there's aldosterone binding to this. And I got pictures of this coming, but I'm kind of having fun with my little markers here. But anyway, but the point of this is the apex, the apices. This is the basolateral surface. Sometimes the apices is called the luminal surface because it connects to a lumen. Who's the lumen? The distal part of the distal convoluted tubules are the collecting ducts. There's no lumen to connect to on the basolateral surface. All right, so the ENaC channels, cares about them. Well, they are the only channels that are involved with the transport of sodium. So this is how sodium moves in and out of the filtrate into the interstitium. Basically, when I say interstitium, it's blood. So here's another cartoon of this, much betterly drawn, I would add. So here's an ENaC channel down here. So here is our aldosterone right here. It binds to an MR receptor. This is a principal cell. We have the filtrate. That's going to be the urine, right? Interstitium, there's the blood vessel over here. So the binding stimulates the central dogma to kick into action, and you generate new ENaC channels. And the ENaC channels allow sodium to come rushing. There's a gradient, a concentration gradient. Sodium comes rushing into the cell, and it goes rushing out of the cell. We'll talk about that in a minute. Well, I might as well tell you right now, because this binding also stimulates sodium-potassium ATPase channels to be put in the basolateral surface. So we could have sodium shoot right through the cell into the interstitium, and then, of course, it can get right into the bloodstream, right? All right, so everything I just said, ENaC channels allow sodium to rush from the lumen through the apical surface or the luminal surface into the principal cell, down a concentration. There's also electrical gradient, electrochemical gradient is there as well that we'll look at. But now we got sodium into the cell. How do we get it out? Well, I kind of spilled the beans on that one. So the binding also stimulates the making of sodium-potassium ATPase transporters on the basolateral surface. So now it can be pumped right through, as I already showed you. The sodium enters the renal interstitium, and it's sucked up by the distal renal capillary, as we've already learned. And yeah. Right. Also a nice question, what's the rate limiting step of the conservation of sodium with regard to getting sodium out of the kidney? What's the rate limiting step? This is the rate limiting step. It is the insertion of the sodium-potassium ATPases. That's the rate limiting step into the basolateral surface. So that's always a nice little board, or maybe my question. I'm sure Dr. Doe or somebody's already hit you with that question before, but there it is again. Here's another little cartoon. You can see the lumen. We got sodium accumulated here. It's anxious to get out. There's a concentration. There's more sodium outside than there is in. So any channel, it'd be happy to go through. There's also too many positive ions out here, and it pushes it through as well. Here's just another cartoon of aldosterone binding to the cell. And the binding causes the expression. What's that mean, expression? That means the making of. It makes these channels. So the channels are made, sodium comes in, and then sodium gets out by these sodium-potassium ATPases into the interstitium and into the blood. And who follows sodium, of course? Water, right? Water. We'll talk about the water here in a second. Right, just another look at it, if you will. Binding, I don't need to go over this again. Oh, this is the electrochemical gradient. So interstitium, so the tubular lumen. So there is lots of positive charge out here, positive and positive. Does that attract or repel? Positive and positive repel each other. So it's happy to get to a more negative environment here. Water follows sodium, as we said, wherever it goes. Not all the time, but usually it does. So does chloride. We never really talk about chloride, but chloride follows the sodium, just like water does. And so we use channels called aquaporins channels. Specifically, probably learned that in high school, but specifically, there's an aquaporins 2 channel that is on the apical surface. Aquaporins 3 and 4 hit the door. That's the door to the interstitium. 3 and 4 hit the door, those are on the basolateral surface. Okay, so, and then of course, what's the principle of retaining sodium, or what's the effect of retaining sodium? Well, water will follow and increase the blood volume, and that will increase the blood pressure. Right, here's another little cartoon here, and we have sodium coming in here, but the key of this one is there's an aquaporins 2 channel coming in, and then aquaporins 2 is going out as well. I believe we have slides coming down the road, kind of scattered. I'm trying to group my aldosterone slides, but I believe aldosterone causes the laying down of aquaporins 2 channels, and after aldosterone is gone, it picks these channels up, and they're not there anymore. The aquaporins 3 and 4 channels are always there, so they're always ready to go, but it's the aquaporins 2 channels are the ones that's regulated. All right, what about potassium? We should talk about that for a second. So as sodium comes into the principal cell, hydrogen ion and potassium both get the boot out of the cell. So how does potassium get out of our principal cell? There's 2 channels that potassium gets kicked out and into the urine, into the filtrate. One are called ROMK channels, stand for renal outer medullary potassium channels, and high conductance big potassium channels, BK channels, from Guyton board books, right? So make sure you know these. And let's take a look at the picture. So the binding of aldosterone, there's our aldosterone binding to an MR receptor, in addition to all that other stuff we talked about. It creates the formation of ROMK channels and BK channels. So now we have potassium moving through these channels and getting kicked out in exchange. We got a positive sodium going this way. We got a positive potassium going that way. So there's a nice little trade-off there. All right, what else do I need to say about that? I think that's all the deeper we're going to get onto that. So we talked about ENaC channels, hydrogen potassium ATPase pumps. Aquaporins channels are not drawn, but aquaporins 2 is here. Aquaporins 3 and 4 is in the back side. So we talked about what else do we know? I think that's good. What about hydrogen? Now hydrogen, I like to ask this question. Hydrogen does not go through a principal cell, right? You learned that. It does not go through a principal. It goes through an alpha intercalated cell. So a little twist there. So these live in the same region, kind of scattered amongst the principal cells. And their job is to excrete hydrogen ion into the lumen through the hydrogen potassium ATPase specifically. That's how they go. Aldosterone's binding stimulates the creation of these hydrogen ion pumps or hydrogen potassium pumps. It also makes hydrogen potassium pumps, and it also makes hydrogen ATPases. There's 2 different mechanisms in these alpha intercalated cells that can go. And of course, this is going to slightly increase the blood pH once all this hydrogen ion gets in there. Let's look at an alpha intercalated cell here. There it is. Principal cell may be down here. So we got the binding of aldosterone right here, delivered through the blood, swam through the interstitium, bound to its MR receptor here. And it causes central dogma to lay down some hydrogen potassium pump, as well as, I keep saying that hydrogen potassium pump is here, and then just a hydrogen ATPase pump. But they both kick hydrogen out in exchange for sodium ion. Remember, potassium and hydrogen get the boot. Sodium, water get retained. And there's just kind of a busy, nice picture that kind of shows everything going on here. Sodium potassium pump here. Pretty much explained all that. Now, aldosterone, in addition to the kidney, it does have some other effects on the body. And we're not going to go crazy, But if you think of the intestine, here's a piece of intestine. If you think of the intestine as the nephron, the lumen of the nephron, or the distal convoluted tubule, or the distal convoluted tubule, or the collecting duct, it's kind of the same deal. Here's an enterocyte. Just think of that as a principal cell. It's the same kind of principle. So we can suck water with aldosterone. We suck sodium and water out of the fecal material. So there's some fecal material, and there's a lot of fluid in here. I guess I should have changed to blue. A lot of fluid. There's the fecal material. This is the intestine. Yeah, it does the same thing. So it sucks the water out, and it sucks the sodium out of this as well. Okay, so really simple as that. And in exchange for, if sodium is coming in, potassium is going out, and so is hydrogen ion. Those are both going out. There's hydrogen ion, and there's potassium ion, and there's sodium coming into the interstitium. And of course, we know the lamina propria has blood vessels here where it can go right in. All right, so that's the deal there. So aldosterone binds to epithelial cells, specifically enterocytes and colonocytes in the small intestine and the ascending colon. The binding causes a binding of aldosterone to these cells, does the same thing, a reabsorption of salt and water that goes into the bloodstream, and it kicks out potassium and hydrogen ion in exchange for that. Did I put that? I'm not sure if I put that in. No, I didn't put that in here, but it does. So, but people with hypoaldosteronism, what is hypoaldosteronism? Well, hypo means it's not enough aldosterone, so they're not secreting aldosterone enough. One of the clinical presentations of that is diarrhea. Why? Because we need aldosterone to suck excess water out of the intestine and out of the colon. Okay, so if you don't have it there, if they're not sucking water, you're going to get diarrhea. There's some other glands too. Aldosterone can stimulate salivary glands and sweat glands to do about the same thing. They retain salt and water. So people with hyperaldosteronism, for example, now they're retaining too much, right? If there's too much aldosterone, you're going to retain or conserve too much sodium and water. You're going to kick too much potassium and hydrogen ion out. But if you conserve too much salt in water, people don't sweat very good. And in fact, that's, some of you people who have problems with sweating, that might sound great. But people can die of this. They have to be really careful of heat stroke who have hypohidrosis, who don't sweat enough. Cystic fibrosis, I think we'll talk about that later, but that's the opposite. There's an opposite mechanism, which we can talk about. They have super salty sweat. Usually the mom is the first one to discover the baby has this fatal disease, cystic fibrosis. Kids live to about 30 years if they're lucky. But yeah, their sweat is super salty, and the moms are usually the first ones because they kiss their baby and their baby is salty, and that's not a good sign. Hyperkalemia, what effect does that have on aldosterone? What's that, hyperkalemia? That's potassium, too much potassium. What effect does that have? Well, heart won't dig into that too much, but if you have tiny increases in potassium, the body does everything in its power to get rid of potassium. Remember, that'll turn on aldosterone more than anything else, so it's hard to get that. But if you do have small increases in potassium level, it can give you some pretty bad arrhythmias of the heart, from palpitations to some of the supraventricular tachycardias. If there's a huge amount of potassium in the bloodstream, it can kill you. Ventricular fibrillation, it's an arrhythmia, but it's a ventricular arrhythmia, and that can kill you. Increased serum potassium, what does it do to aldosterone? Well, increased potassium concentration or hyperkalemia, as I just said, that is the strongest stimulator for the release of aldosterone. And the glomerulosa cells have receptors for potassium ion. It's a stronger stimulator than angiotensin II and stronger than ACTH. All right, in response to increased levels of potassium, potassium binds directly to glomerulosa cells, as I said, strongly stimulates the release of aldosterone, and we're experts on aldosterone now. That hits the principal cells of the kidneys and causes the conservation of salt. Water comes through the alpha intercalated cells, so you got the conservation of salt in water, and that way, then we got two molecules going the opposite direction, hydrogen ion, and there's our excess of potassium right there. Kick it right out into the future urine, into the filtrate. Here's another little thing I drew before. Just showing you this is, you definitely know this stuff, right? But here again, so here's the urine going down here. We'll say this is the connecting tubule, or maybe the proximal collecting duct, whatever. And the blue little balls represent the incomers. They're conserved by the binding of aldosterone, sodium, and water. And then the ones who get kicked out are potassium and hydrogen ion are in red. And so you see aldosterone binding to its MR receptor, and that via the mechanisms and the transporters we talked about. Bottom line, potassium, hydrogen kicked out into the urine, into the filtrate, and exchanged for sodium and water. Again, basolateral surface is here. Apical surface is here, sometimes called luminal surface. Why? Because it's bound to the lumen, or very close proximity to the lumen. How does potassium get out? Well, we know, or did I talk? Yeah, we talked about this already. So potassium, did I talk? Can't remember if I did. Yeah, I did. Stimulates the expression of sodium, potassium, ATPase pumps. No, we didn't. We talked about sodium getting out, but the potassium can use the pumps on the basolateral surface of the principal cells, so on the bottom. The principal cells, we already said where they live, but they live in the distal convoluted tubules. Guyton calls these connecting tubules. Physiologists tend to call these, anatomists don't, but physiologists, Sherwood calls them connecting tubules, another big physiology book. So then sodium rushes down a concentration gradient into the principal cell. So great, but now we got to get potassium out. So the increased potassium again stimulates the expression of another kind of channel. These are called ROMK channels. ROMK potassium channels. These are on the apical surface of the principal cells. This is their way out. So now we have a way in and a way out of the principal cell. So potassium can move that way down its concentration gradient. Do I have a picture? Oh, I thought I had a picture. Where's the ROMK channel? There it is. There's a ROMK channel right there in this little cartoon. So yet another one. So, geez, aldosterone binding cranks up central dogma, cranks up the ribosomes, right, to make all these channels. So ENaC channel, ROMK channel, BK channel. Pretty busy. All right. Sodium regulation. Again, my iPhone's not here, so I have no idea how. Probably can do a little more. It feels like we could do a little more. So let's do some aldosterone, sodium regulation. So aldosterone, of course, we'll get back to aldosterone. That's the principal regular body sodium. We already know that. But sodium can't. Here's something you may not know, and I ask this question often, so make sure you know the answer to this question. Sodium, even though aldosterone, which is secreted from, who else secretes aldosterone? glomerulosa cells, even though it's secreted by glomerulosa cells, and even though aldosterone is the principal regulator of sodium, you would think sodium could bind directly to glomerulosa cells. But it doesn't. It's really strange. So sodium does not directly, it doesn't bind, period. I could take directly out of there. It doesn't bind to glomerulosa cells. So how do you get rid of it? What if there's too much of it? How do you regulate it? Well, let's talk about how to get rid of it. You just had a Big Mac put with salty fries, and now you got too much salt. How do you get rid of that salt? Well, no, the sodium can't bind to aldosterone or can't bind to the glomerulosa cells. So there's got to be another mechanism. So this is a very complicated and long process, but we can boil it down. We can kind of cut to the facts. So the excess salt that you just swallowed, water the fluid of your blood and the fluid around your cells and the fluid inside your cells, they will not tolerate an offness of sodium. And so once the sodium gets into your blood and starts getting into the interstitium, the cells will release water to neutralize it and bring it back to its proper concentration levels. Great. So in other words, water is going to go into your interstitium. You get a little puffy maybe, and then water is going to go into your bloodstream as well. So bottom line, you're going to have a little hyperkalemia, and your blood pressure is going to rise a little bit. That's the mechanism right there. Right? Water will soak out of the cells everything I said. So the increased blood pressure does two things. And we can boil this down to two simple things to answer this question. It's going to turn off the R2A system. And I mean, I could go into all the mechanisms, but basically the juxtaglomerular cells are not going to sense hypo pressure anymore. They're going to sense pressure that's too high. That shuts them right down. And we could go right through the gamut. The apple pickers, now the belt is going to be sped up, so they're not going to pick as much sodium out. So macula densa is not going to freak out, et cetera, et cetera, et cetera. We could go through those 9 things that we talked about last time. But just in case, for whatever reason, if that doesn't work, we have another system we haven't talked about. That's the natriuretic peptide system, or just the natriuretic system for short. That's the natriuretic peptide and the BNP and the CNP. Remember those things? Those do the opposite of the R2A system. So both of those systems are affected by too much salt in your blood. And the bottom line of the effect of the R2A system, if you turn it off, there goes your aldosterone secretion. Well, if you don't secrete aldosterone, you can't reabsorb sodium. So any sodium in the filtrate, it's not going to kick sodium out, but it's certainly not going to take sodium out of the urine, because now we got to get sodium in the urine. That's how we get it out, right? We have to pee it out. So that mechanism is shut off. Okay, and then more sodium is pushed into the filtrate because we said angiotensin II can actually cause the potato pickers to pick more sodium molecules out of the filtrate. So that system is shut down. The conveyor belt's going faster, and now the potato pickers aren't of the proximal convoluted tubules. They're not affected. So they're going to let more sodium dump into the filtrate. And macula densa is not going to care. It's happy. It only freaks out if the sodium gets low. So now you're starting to urinate out sodium, and that's going to eventually level things off. And if that isn't enough, the natriuretic system does just kind of a duplication, but it's the same thing. The ANP binds to glomerulosa cells, and it shuts off the production of aldosterone. So we can't suck sodium out of the water. It's a double mechanism. It also binds to the proximal convoluted tubules, and it stops the absorption of sodium from there as well. So it actually, I think it stimulates this for more sodium to be dumped, because it's the opposite of angiotensin II. right? So that's the bottom line. So there are other ways you can lose sodium as well. So you could go sweating like crazy. You could sweat it out through your sweat glands. You could lose sodium, but that's not going to happen. Cystic fibrosis, you're not going to go get cystic fibrosis to fight this. So that's not really a way that can do anything. You could give yourself diarrhea, and that could get rid of salt and water because it can't be reabsorbed into the intestinal wall, but you're not going to do that. So these are not viable ways to get rid of sodium. Yeah, it's just you turn off aldosterone is the main thing. Pop quiz. Who can bind to glomerulosa cells? Who can bind to glomerulosa cells? Let's see. Angiotensin II, can that bind? Absolutely. Potassium ion, yeah, that binds more readily than angiotensin II. ACTH, adrenocorticotropic hormone, yeah, it can bind as well. Not super, super strong. Sodium ion, nope, can't do it. All right, hyperaldosteronism, I think should we stop it right there? I think that's probably a good place. We went a little long last time. This is a good place to stop. I'll count the slides and maybe I'll pick up. If not, we'll pick up on this next week. All right, see you in the next lecture.